synthesis, biochemical characterization, and antibacterial activity of Dioscorea pentaphylla. I am Dr. M. Suriyavathana, professor in the Department of Biochemistry, Periyar University, Tamil Nadu, India. This is very interesting and a fascinating concept about this tuber called Dioscorea pentaphylla, which contains a versatile nutritional potential, and it is considered to the family of tubers. Dioscorea pentaphylla is also known as the five-leaf yam, is a climber, and it is a native to that of the tropical regions of Asia. Its tuber, which resembles potato ham, has been traditionally used for food and also for the medicinal purposes. Just go on to the nutritional profile of Dioscorea pentaphylla, which has been enriched with carbohydrates abundant in fiber, and it contains a potential source of vitamins and minerals, and also the serving as antioxidant powerhouse. Dioscorea pentaphylla is a good source of carbohydrates, whereby it provides energy and also fueling the body by metabolic process. Besides this, it has a high fiber content which promotes the digestion and also regulates blood glucose levels and aids in the defense called the immune safety. This also enhances contained amount of the tuber valuable for the rich source of vitamins, especially for vitamin C and minerals like potassium and magnesium. Dioscorea pentaphylla, which has a rich source of antioxidants, which provides an antioxidant against the free radical defense and also overcomes the oxidative stress and protects against the cellular damage. And exactly this carbohydrate and fiber content, what we assess from this particular tuber, carbohydrate goes rarely in the range of 17.5 grams per 100 grams of this tuber, and dietary fiber nearly is 5.6 grams per 100 grams. Then vitamins and mineral compositions, which will specify its enrichment of the vitamin and mineral potential, especially on vitamin C. Dioscorea pentaphylla is a good source of vitamin, whereby it provides antioxidant support because the vitamin C is going to act as a non-enzymatic antioxidants to give the free radical scavenging. Thereby, it supports the system and also immune function and is essential for the collagen production. Magnesium is an essential for both bone and also muscle relaxation, and also essential as an coenzymic component for the energy production. Potassium, it regulates the blood pressure and it's involved in that of the nerve impulse and also transmission process. Other minerals or the contains in the tuber is that zinc, which is also very much essential for that of the body, immense regulatory function. Here we are going to focus on that of this enriched tuber contains so much biochemical and nutrient potential. We have worked on to work out for that of the metallic nanoparticle synthesis and the metallic nanoparticles green synthesis, which is used for many diverse medicinal plants, and one among them is which contains the therapeutic properties. Herbal medicine is also known as the botanical medicine, or otherwise we can define it as the phytomedicine. Dioscorea pentaphylla is a species of the flowering plant in the yam family, known as the common name five-leaf yam. In local name, in Tamil, we used to call it as irula kilang. The phytocomponents which have been analyzed and found out, which are said to be the secondary metabolites, they are the health beneficiaries which is going to serve as the phytomedicines for various health issues we'll be working on. These phytochemical substances are presented in that of the nutritional herbal medicines. Flavonoids are many important, and other phenolic compounds have an effective antioxidant property, anti-cancer, antibacterial, cardioprotective, anti-inflammatory, and also provide immune function and promotes the protection against the UV radiation, and especially for the skin, an interesting candidate for pharmaceutical and medicinal application. We've just gone to the green synthesis of the silver nanoparticles using Dioscorea pentaphylla rhizome. Characterization of the green synthesized root of this Dioscorea pentaphylla rhizome. And we are later on, after synthesizing the nanoparticles by using the Dioscorea tuber extract, then we went on to see for the characterization by GCMS analysis, and also we went on to go for the assessment of the biochemical potential of the Dioscorea pentaphylla extract. In that, we have analyzed for that of the polysaccharides and content of the protein and the cholesterol. And parallelly, we assess the antibacterial activity of the Dioscorea pentaphylla rhizome extract by using against the organisms called E. coli and also Bacillus subtilis by two methods, one by four-plate method and another by that of the cup diffusion or zone of inhibition. The collection of this plant is from that of the nearby area forest called Bomidi, which has been identified and authenticated. And the rhizomes of this pentaphylla were washed with water, shade dried, and the temperature, room temperature, and we are powdered by an electrical blender. And the scientific classification or taxonomy of this Dioscorea pentaphylla, it belongs to the family Dioscoreae, genus Dioscorea, species Dioscorea pentaphylla. This is the specific structure of this particular root tuber on what we have been working on. And this is the aerial part of this plant, and this is the root tuber, which is going to have all the nutri-potential. And this is the aerial, it is a tuber. And how this extraction has been assessed, and the dried root tubers are then is powdered, and later on this powdered rhizome powder are then allowed for the extraction by using the soft-sled apparatus. For this, we have used 20 gram of the dried powder and of the rhizome, and we will analyze with that of in the soft-sled apparatus using different solvents, various solvents, namely aqueous, ethanolic, chloroform extracts. And after that, this extract has been used for that of the biochemical assessment for the estimation of the polysaccharides and protein and cholesterol, and also parallelly estimated the iron content, calcium, and non-enzymatic antioxidants, namely vitamin A and vitamin C. And this exactly, which is few of that of the green synthesis or the silver nanoparticles synthesized from that of the Dioscorea pentaphylla rhizome. And this has been dried, and later on it has been powdered. Now it has been added to that of the 900 ml of the silver nitrate solution, and this has been mixed with that of the rhizome powder, whether it is AgNO3 pentaphylla rhizome extract. Then after adding of this powder to this of the silver nitrate, it undergoes the reduction, and thereby there is a change in the transformation of the color. Now the synthesis initiated, whereby we go for the AgNPS, otherwise called as the green synthesis of the Dioscorea pentaphylla. And the antibacterial activity was evaluated using against this of the two organisms, namely the common microbial species, bacterial species, namely E. coli and Bacillus. These strains are provided from the department, and they have been subcultured, and we have been made from the Department of Microbiology, and we have used for this study. And the standard drug against this bacterial infection has been studied with the used is called the tetracycline as served as a standard. And the medium we have used for our antibacterial study is Muller-Hinton agar, following the techniques by four-plate and also cup diffusion method. Then to analyze the stability of the nanoparticles once synthesized, the visual examination has been confirmed by the synthesized nanoparticle, and the color of the mixture distinctly changed from greenish yellow to brown. So this is what the pictures to representation, how the nanosynthesis has been initiated. Hence, the green synthesis of the Dioscorea pentaphylla rhizome extract, it potentiates as a reducing and a stabilizing agent for the formation of the AgNPS. Biochemical assessment, estimation of polysaccharides. Among the three different extracts we have used, aqueous, ethanol, and chloroform. Aqueous holded the highest content of the polysaccharides, namely 4.75. With that, when compared, next follows will be ethanolic extract, and finally with that of the chloroform. And this is about the protein content of the Dioscorea biochemical component, which has been quantified. Here also, the aqueous extract holds the highest protein content when compared to that of the ethanol and the chloroform. Then cholesterol, this Dioscorea possesses the high amount of cholesterol was been notified in chloroform extract, followed by that of the aqueous and the ethanolic extract. And this is the estimation of the polysaccharides, protein, and cholesterol in the different solvent extracts. And in this case, we can specify that the Dioscorea pentaphylla aqueous extract upholds high content of biochemical constituents, namely polysaccharides, proteins, and also meager amount of cholesterol, little cholesterol. and its estimation of iron content present in Dioscorea pentaphylla rhizomes. And when compared to that of the aqueous ethanol and chloroform extract, it possessed high content of the iron in aqueous extract. Followed similarly, the estimation of calcium. We happen to notify that high content of calcium mainly when present in Dioscorea aqueous extract rather than with that of the ethanolic and the chloroform extract. This is the comparative concept about the iron calcium levels of the Dioscorea using 3 different solvents. So in all, both calcium and iron content is being admirably available rich in the aqueous extract followed by that of the ethanolic extract. Then estimation of vitamin A, that is antioxidant vitamin A and C. Here also vitamin A content is notified to be higher in aqueous, followed by that of the ethanol and also the chloroform. And in case of vitamin C, we happen to see that there is a content high level of vitamin C in ethanolic extract, and next will follow this here aqueous, followed by that of the chloroform. So the synthesis of the green nanoparticles, silver nanoparticles from the Dioscorea pentaphylla extract, the brown change in the color, that is the reduction of the silver, the brown color was due to the surface plasmon resonance excitation of this silver nitrate, that is the AgNPS. So the sample is exhibited, thereby there is a variation in the change in the UV length that is called as the spectrum, reducing from 250 to 475. So this graph will represent the result initiation of the synthesis of the reduction initiated by that because of the surface plasmon resonance. And followed that the spectral of this plant extract, the peaks ranging from 520 to this one 340 3407 OD, that will represent the FTAR. That is this particular characterization provides the abundance of the functional groups presented in that of your synthesized nanosynthesis Dioscorea pentaflora particles. And weak peaks ascribed to that of the unsaturated carbon-carbon bonds. And sharp peak which provides that of the alkenes, and also it provides a high content of the carbon source. The frequencies of the range of AgNPS Dioscorea pentaphylla rhizome extract, and this gives the peak position and the type of the functional groups and the class of the compounds available. So when you see that it contains a good store of alkaline, alkene, carbon alcohol, primary alcohol, and also halo compound. So the bonding at the peak designation will provide you the strong bondage for alcohol stretching and also for that of the primary alcohol. So mostly it is encompassed with that of the alcoholic groups as such in the characterization process. And this picture will provide you the information about the scanning electron micrograph, that is some structural installation of the AgNPS of Dioscorea pentaphylla extract. The result observation reveals that is they observed a structural morphology of the AgNPS, that is silver nanoparticles, which has an average particle size varied in the length of 12 mm. And thereby the SEM analysis and outlay mostly all the structures resembles a cuboidal and spherical structures after the nanosynthesis. And this results of the antibacterial activity of Dioscorea pentaphylla rhizome extract, and we will check with that of the E. coli and Bacillus subtilis by two approach, one by 4-plate method, and we have served this first one served as a control plate, and we have the organism along with that of the extract and the source. So the antibacterial activity of Dioscorea pentaphylla in the ethanolic extract exactly specifies there is no growth, so which has been having a potential antibacterial effect. And also we check for that of the next organism called the Bacillus subtilis in the chloroform extract, ethanol, and also for the aqueous extract. And this provides the activity of the Dioscorea antibacterial activity against that is organism called the E. coli and Bacillus by a zone of inhibition. Otherwise, we can call it as the cup diffusion method. It depends upon the concentration. We have checked with that of different or in the varying concentration, namely from 25 ml, 50, 75, and 100 ml. So we see the zone of inhibition depending upon the increase in concentration. which also confirms the effectiveness of the antibacterial potential imbibed in this particular tuber source. To sum up the entire process of the work, the amount of the from the Dioscorea and pentaphylla, the amount of the total carbohydrate present in this tuber or rhizome, which contains a very good potential source of polysaccharides especially. So these polysaccharides are mainly energy boosters or the source of food, especially for the of the low socioeconomic groups who depends on this particular food process. Next is that the protein content of this extract also this seems to be very much effective, and it contains a notable amount compared to that of the carbohydrate. And parallelly, it contains a very little amount of there is what called the lipids. What we have estimated is the cholesterol. Besides this primary nutrients, namely carbohydrates, protein, lipids, it contains a very enriched level of both vitamins and minerals, mainly the minerals calcium and iron. Calcium is essential for bone growth development and also for the muscle excitation and also muscular function. And we know that especially iron is for that of the normal RBC level to give the normal blood function as well. So the bioreduction on the nanosynthesis initiated, which has been notified by the UV spectral sharp peak and a stable peak obtained at 250 to 475, which confirms that silver nanoparticles has been effectively synthesized from this plant source bioreduction process. Followed by that of the characterization done with that of UV FTIR. This reveals that it contains a wide range of functional compounds, namely carboxylic acids, alkenes, alcohol, organic acids, and other compounds and halogens, etc. And the results of the scanning electron microprocessor analysis, which has provided a cuboidal shape of the spherical structural morphology of the green synthesized AgNPS of the pentaphylla. Dioscorea pentaphylla rhizome hold a good store of antioxidants, especially vitamin C, and followed by that of vitamin E. Besides this, providing the nutritional potential, namely the rich nutrient content and also the carbohydrate as a biochemical component of nutritional aspect. This is containing a very important component aspect of that immunity that's called as the effective against the phytomedicine, where we can recommend later because of the results provided from the antibacterial study, which is serving as an effective inhibitory activity against the selected strains, namely E. coli and also Bacillus subtilis. To conclude, the research has been comprehensively focused in order to explore the biochemical potential of Dioscorea pentaphylla rhizome in the gillette, antibacterial efficacy against the infectious pathogens, namely the E. coli and Bacillus. Dioscorea pentaphylla rhizome has been screened and has been completely assessed. And this rhizome has an effective nutrient potential source with abundance of vital nutrients and has a remarkable energy resource along with that of the essential minerals, namely iron and calcium, whereby it will certainly serve as a potential natural antioxidant source with the margin of safety. The recommendations, what I suggest according to this particular piece of work has been carried out in our lab. This tuber is containing a high risk recommend for that of the malnourished and low socioeconomic group as a vital nutripotential source and will certainly serve as an effective energy source in the future. And this is a small cap of my plant therapeutics laboratory at Periyar University. We focus on with that of the working with that of natural compounds, which are considered to be the golden standard to mark the outstanding phenomena of symbiosis, which recommends and encourages the use of traditional herbs, natural remedies in the healthcare sector due to easy availability. Another thing is the affordability. Herbal medicine involves alleviations of disease by plant-based formulation method. And our mission of our lab is to traditional insight embroiled in indigenous system of medicine needs scientific validation, especially in its chemical parameters. And this is the process we are undertaking in our lab, what we do for nearly 20 years in the phytochemical lab, that is the phytotherapeutics lab. And yearly we have screened and assessed and formulated number of compounds for different clinical manifestations. And we go on to this process. We work on all aspects of this with herbal formulation, nanosynthesis, characterize, purify compound. Then we go for the clinical manifestations, and also we do with the in silico study for this particular all classes of plants. And these are the different plant sources which I have been working on nearly for 20 years on different herbal medicines we have been successfully got out from our laboratory. And these are my publications on the books on which I will record the nutraceuticals, bioefficacy of nutraceuticals, and these are the published books available. And this is a patent I have been designed from the herbal system for that of the drinking water process. And this is our happy to share our lab scholars along with that in that office. Save nature to sustain bioscience in work. Thank you. Thank you for your patience hearing. Any questions?